Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest who is involved in creating a better tomorrow on many unique fronts and who wears many different hats. And I just want to take a little time to go into this bio today. We have the honor of being joined by Dr. Michael Hufford, who is the co-founder and CEO of Ligenesis, which is a clinical stage cell therapy company that's focused on transforming patients' lymph nodes into bioreactors capable of growing functional ectopic organs. He is also serving as the interim CEO of Morphoceuticals, a company that's focused on modifying electric potentials in cells and tissues for a variety of applications in regenerative medicine. In addition to that role, he also serves as a scientific advisor to Juvenescence, a biotech holding company focused on developing therapies and products to help modify aging and help people live healthier for longer. An entrepreneur and drug developer, Dr. Hufford has over 2 decades of experience in the development of FDA regulatory approved products, including small molecules, biologics, as well as drug delivery technologies. He's designed and executed clinical trials and drug development programs across a wide range of therapeutic areas, from orphan diseases to complex psychiatric and oncology indications. He has broad experience in and out licensing of both preclinical and clinical stage assets involving complex corporate partnering deals, as well as performing investor relations, raising money in the public and private markets. He also is involved in philanthropic work, including co-founding and serving as the CEO of Harm Reduction Therapeutics, which is a really cool nonprofit pharma company developing a low-cost, over-the-counter intranasal naloxone. Dr. Hufford earned his undergraduate degree with distinction from Purdue University. He did his master's and doctoral degrees in clinical psychology from University of Pittsburgh before completing a research and clinical fellowship in the Department of Psychiatry at Harvard Medical School. He has hundreds of scientific publications, presentations, and op-eds over the years. A lot of really interesting themes to get into today with him. Dr. Michael Hufford, welcome to our show, and thanks so much for joining us.
Oh, thanks so much for having me. If there's anything more painful than hearing your own biography, I'd like to know what it is. So I'm glad we got through that. Thanks. It's good to be here.
It's impressive, Mike. And I jokingly, I mean, you're like the Energizer bunny of bioentrepreneurship here. Before we get into some of these companies and some of the themes that you're involved in here, I'd love to just, as we typically do, sort of give you the mic for a few minutes just to talk a little bit more about Michael Hufford, a little bit more about your background, where this whole journey started out, and a little bit about how you segued sort of from the whole area of clinical psychology into bioentrepreneurship on steroids.
Yeah, so no, thanks so much. I really appreciate the interest and the questions today. So, my background started off in clinical psychology and got exposed to the drug development industry really through an academic advisor of mine who's doing a lot of consulting for the pharmaceutical industry. And what struck me most about it was at the intersection of science and business, you had the capital to enable work to proceed just so much more rapidly than you could in academia. One of my first academic publications was a book chapter with an advisor of mine, and I just got my bachelor's degree and started graduate school when we submitted it. And by the time it was published, I had my PhD. I didn't get my PhD quickly. It just took that long, right? And so the pace of academia I found very much frustrating. And yet at the same time was obviously deeply in love with the scientific process and the scientific process of discovery, and really saw in the pharmaceutical industry and biopharma in particular, the ability to leverage what's best about science combined with what can be the best about business, which is the sort of singular focus of achieving certain operational objectives. And if that's in the service of bringing new medicines forward, I really couldn't, imagine a more kind of noble enterprise. And so, yeah, for the past 2 decades have just been fortunate. I joke I'm sort of the least interesting person that I know, right? I've just been extraordinarily fortunate to work with some incredible minds, some incredibly creative people, and see technologies forward. And I'm sure we'll have a chance to talk about those. But that's a little bit of the backstory of the transition from kind of a tenure track academic position to a much more industry focused, and then eventually to a much more small company focused, where I think even more so, right, you have the opportunity to have your hands in a lot of different activities and really steer the ship, as it were, toward these really exciting missions and objectives.
Yeah, yeah. And just, for the record, I do not find you the least interesting. I mean, I find you extremely interesting. So yeah,
I'm not the least interesting.
Don't worry about that. Mike, before we get into some of the companies, there's one place I wanted to stop, because I took some time to sort of watch a lot of the interviews that you've given over the years and the presentations, and one theme that you touched on recently when you were giving there was an investor presentation, was this principle of working with nature. We have all these fascinating natural processes, whether it's regeneration, repair, what have you, and earlier on in your career, and you mentioned this, and I thought it'd be interesting just to talk a couple of minutes about it. When you were working in the biologic space with amylin, you worked with this fascinating GLP-1 peptide, which was originally derived from the spit, the salivary secretions of the Gila monster. And I'm very fond of sort of our industry and how the last 100 years or so were built in many ways from clues from nature, whether it's the plant kingdom, whether it's all sorts of interesting little creepy crawly things that gave us a lot of the interesting early biologics. Talk a little bit of that theme for a moment, if you would, because I think it feeds nicely into a lot of what we're going to be touching on with the different companies.
Yeah, the point I was making there was that, just looking back really selfishly, I suppose, at my own career, at where I've had the most success in terms of leveraging biology for therapeutic purposes. And it's always been, exactly as you said, working with instead of against nature. And by that, I just meant, and I harken back to working on Byetta at Amylin Pharmaceuticals, where, so this peptide in the saliva of a Gila monster, right? The Gila monster will not eat for months at a time. When it finally eats, among the peptides it secretes is this GLP-1, which of course now has become this whole, very powerful class of anti-diabetic medicines. But really, nature had engineered that to be so potent that when the structural chemist at Amylin tried to make it more potent, we really weren't able to. I mean, nature had made it really over millions of years to be this incredibly potent, very selective way to affect metabolism, and there really was no way to out-engineer that, at least at the time. Now, of course, we have orally available GLP-1 agonists and what have you, but that was just one example where, when you work with nature, you just encounter far fewer roadblocks, and the very roadblocks that block so many medicines from becoming valuable therapies, right? Off-target toxicities, things that you don't anticipate. And a similar story at Ligenesis that I'm sure we'll be able to talk more about, but there too, leveraging the natural regenerative capacity of the liver, right? Using the lymph node, which naturally is this very powerful bioreactor naturally for T cells, but we're using it for other purposes. But again, sort of tweaking those things can be far more advantageous. And if you think of what are the key problems that stop drugs from making it, it is very often off-target toxicity, right?
Yep.
That is, efficacy is always a challenge, but it's the unforeseeable safety events. And so when you're leveraging biology to do things that it naturally wants to do, And of course, Morphoceuticals and the work out of David Kaplan and Mike Levin's lab at Tufts being a dramatic additional example of that. You can accomplish these remarkable therapeutic feats and do it in a way that avoids the pitfalls that so many other drug development programs fall into.
Absolutely. Absolutely. Yeah, let's with that, let's segue into the companies. And I'd love to start with Ligenesis, because you were just saying, here's the premise. On one hand, this powerful in vivo bioreactor. Secondary, we have livers, which have this really interesting hypertrophic regenerative capability. And you're really merging these two capabilities together for these ectopic organs, growing them from organoids and so forth. And it's very interesting contrast, but we've done shows recently on themes like bioprinting, on extracellular decellularization, and xenotransplant, where you're looking at the big thing. Here we're starting at the other end, right, where we're talking about sort of this morphodynamic growth from the very early stage, the way it naturally happens in embryos and sort of regenerative biology in the liver. Take us a little into the Ligenesis story, if you would.
Oh, and one, I'm going to steal the phrase morphodynamic. I actually love that, so I'm going to start using that now. So just as an orientation, if folks aren't aware of the Ligenesis story. So Ligenesis is a technology that came out of Eric Lagass's lab at the University of Pittsburgh and the McGowan Institute for Regenerative Medicine in particular. And what Eric discovered in his lab is that the lymph node could serve as a very powerful, highly efficient, and very effective bioreactor for a variety of different tissue types, not just T cells, right? So naturally, we have 500 lymph nodes distributed throughout our body. From an evolutionary perspective, we have them to fight local infections so that they can quickly culture, millions upon millions of T cells to help fight infections, do that very, very rapidly. So as you know, right, you can go to sleep feeling a little under the weather, wake up with a head cold. You'll have pronounced nodules underneath your jaw that you're literally feeling the tens, hundreds of millions of T cells that your lymph nodes have acted as a bioreactor to form to help fight that infection, right? And so what Eric found is that he could put a variety of different cell types. He started with the hepatocyte, the liver cell, into a lymph node. And what he found is really just as cancer probes its surroundings to find niches to grow tumors, to continue to expand, just the way that cancer naturally does that and finds the lymph node this incredibly hospitable environment to rapidly form tumors and spread throughout the body, Eric found that you could leverage that same biology for a therapeutic purpose, that if you put hepatocytes in the lymph node, they too would thrive, and they would do what hepatocytes do. Hepatocytes are a very unusual cell insofar as they are a natural stem cell, right? Yep. And when given the pro-growth cues from a diseased liver, so a diseased liver is emitting dozens of biochemical signals to try to get the hepatocytes to grow. But in an end-stage liver disease, it's very fibrotic. Really, the house is falling apart, right? And so the hepatocytes cannot grow the functioning liver lobules and portal triads and central veins that are required to filter the blood in this fibrotic environment. So those pro-growth signals try to heal the liver, but are unable to, but they're circulating throughout our body. And so when you put a hepatocyte within a lymph node, they detect those pro-growth signals, form liver lobules, form central veins, portal triads. We found can also form bile ducts, and will recapitulate within the lymph node. In fact, over time, the lymph node will disappear, and those hepatocytes will form an ectopic liver. That is a small miniature liver outside the natural anatomical spot that the liver should be. So from that remarkable discovery, Eric then went on to show that a variety of different tissue types would use the lymph node in the same way, from pancreatic islet cells that could sense glucose and rescue diabetic mice, to renal fragments where we can grow an ectopic kidney, to the thymus, which has a remarkable therapeutic potential, including potential anti-aging effects, because the thymus, as we age, tends to atrophy. Yep. And so if you could regenerate, and we're working on this right now, actually, if you can put a young thymus in an old body, we have some good evidence now, and in fact, some will be publishing soon, that you can really shift the entire transcriptomic profile of an animal toward a more youthful phenotype. But this just all goes to show that what are we using at its foundation? The platform is the lymph node, this natural bioreactor. And you're absolutely right. As sexy and cool as a lot of the xenotransplant stories are, and the bioprinting, and it's all incredibly cool stuff. If you think of the regulatory burden, and so, part of this is just my background as a drug developer, right? Like, as sexy and cool as the science can be, at the end of the day, I want something that can help patients, which by default means you have to get through the FDA. So when you look at these programs from a regulator's perspective, right? If you're grafting a few hundreds of millions of cells, right, it is a much less great immune burden relative to a full organ transplant. It is a far easier case for our lead first generation therapies are all allogeneic, so they're cadaverically derived cell therapies, but we've been transplanting organs for decades and decades now, right? So there's a lot of regulatory precedent for that. We understand the immune suppression burden. If you contrast that to xenografts, which also have a many decades long history, a lot of people aren't aware of all the xenograft work that was done in the 60s and another resurgence in the 90s, and we're seeing another resurgence now about 30 years later. There's just a much more complicated, right, immune burden profile with those technologies. Where with the lymph node, you have these distributed throughout the body. They efficiently bioreact a variety of different tissue types. We also use an endoscopic ultrasound. So we go through the mouth of the patient to engraft lymph nodes adjacent to the liver so they get the strongest pro-growth signals possible. But that also means it's a outpatient, very low risk medical procedure. So again, as a drug developer, I'm like, how do we decrease risk? And if you're engrafting a whole organ or a whole pig organ or a bioprinted organ, there's all sorts of concerns, right? Around rejection, around vascularization. It's not that those aren't eventually going to be tractable, but they are complexities. And if your interest is, getting things through the FDA, you're looking for really the path of least resistance, right? And so for us, allogeneic cells, tremendous regulatory precedent for those. The immune burden associated with those much less than full organ transplant, certainly orders of magnitudes less than xenotransplant. And then we have this ease, this route of administration, which also decreases the risk, right? A lot of the patients in our first phase 2A clinical trial, these are patients who have been declined transplant, largely because they're not well enough to survive the procedure. And so again, if you were transplanting them with a pig organ, you would have an even higher hurdle, right? Not to mention all of the costs associated with full transplant, of course. So what we're trying to do is help patients that are sick enough that they don't qualify for the transplant list for one reason or another. But the endoscopic ultrasound, again, is that path of least resistance. It's a safer way to engraft these cells. And so I admittedly have a sort of, almost mechanistic and, way of looking at drug development assets. And it's really about ultimately kind of making your own life easier, largely because that's what it takes to convince an FDA regulator that the benefits of the therapy outweigh the risks, right? And that's always what their concern is. I graph this on the board sometime, a teeter-totter of risks and benefits, but I remind the audience from the FDA's perspective, the fulcrum is not in the middle of the teeter-totter. It's way skewed over, right, toward the safety side. So that any hint of safety concern craters a program, and only a remarkable amount of benefit is required to offset even relatively slight risks. And so when I look at these assets, I always think, okay, what is going to have the risk-benefit profile that's going to help convince the agency that, and ultimately physicians and patients, that this makes sense. So we're targeting patients for our lead indication at Ligenesis with end-stage liver disease, who are otherwise declined for transplant, so they're really just looking at palliative care. They don't have a lot of other options. We're engrafting just a few hundred millions of cells, so the immune suppression burden is much less. And so that's another example, I guess, of really trying to work with nature and make the risk-benefit profile really as compelling as possible to regulate.
The other thing I found in your recent presentation was really cool because, aside from the science and then the regulatory, you actually took the presentation a little into the commercial side, how because of what you were just mentioning, you don't require a tremendous amount of facilities all over the place. I mean, you can really, with a small sort of footprint, supply the market. Say a couple words about that, if you would, because I think that's something that's overlooked here.
Yeah, it's certainly not the sexy part of the story, but if you think of how do you get capital to flow into a drug development space, you really have to be able to tell a compelling commercial story of how are you going to make money, right? And so not only is having a life-saving effect and growing an ectopic organ an incredibly important medical advance, but the way in which we do it, I think, is extraordinarily compelling just on the commercial aspect of it. So let's talk about that for a second, right? One is each donated organ. So each donated liver, let's take that as our lead indication. Each donated liver is made up of billions of hepatocytes. And because we're only engrafting a few hundreds of millions of hepatocytes, the math just works in our favor, right? Each donated organ could easily treat 75 or more patients. So if you want to talk about like a classic pitch of a startup of disrupting something, like that is truly disrupting the supply-demand calculus when it comes to organ availability. I mean,
absolutely.
Every day in this country, I mean, today, right? A couple of dozen people are going to die waiting for organs that did not become available. And that's fundamentally a supply-demand problem, right? The demand far outstrips supply. Using this technology where one donated organ, you actually probably won't be able logistically to arrange that many engraftments in time, right? Like talk about a good problem to have. We're upending the supply-demand so much that a single donated organ can treat up to 75 patients. Means your primary problem is a logistics one, which is a fantastic problem to have, right? So on the one hand, we're also using discarded organs. So instead of a one-to-one treating one organ, one patient with one organ, we can treat 75 patients with one organ. Two, those organs are donated, but otherwise going to be discarded. So in this country, the percentages vary. Let's say conservatively 10% of donated livers are discarded, right? And so they can be discarded for any number of different reasons. Oftentimes, a transplant surgeon will look at it, say, oh, it's not quite right for this patient. But for our process, because we're going to enzymatically digest that organ and isolate the key hepatocytes within it, we can use those organs. So it's a relatively inexpensive cell source. And then to your point that I was making in a previous presentation, this really is a bolt-on capability to any existing GMP facility, right? So if you think of CAR T therapies, you think of CRISPR, you think of all these technologies, companies have invested in universities as well, oftentimes hundreds of millions of dollars in these GMP facilities. Oftentimes they're enormous suites because you need that space and a lot of complicated infrastructure to pull off the genetic engineering for some of these recent cell therapies. That's all great. My point is, we can actually fit in the corner of one of those. I need 10 feet of bench space and a hood, right? It's a technically complex process. It's over 70 steps. It takes about 6 hours. You have to have trained people doing it. It's all part of our own CMC package with the FDA. But that being said, relative to CRISPR or CAR T, right, what we're doing is relatively straightforward. We're not genetically manipulating the cells. We're really mechanically and chemically isolating them in a very specific way from the donated organ. But that means that each liver isolation costs about $15,000 in reagents. And if you remember what I said about 75 patients per liver, you start dividing by the 15,000. I mean, like I tell folks, the cost of goods for our cell therapy, I mean, it's going to have software-like margins, right? And this is, at the end of the day, just on the very unsexy business side of this, relative to say, implementing CRISPR in a pig, raising that pig on a GMP farm throughout its life, and harvesting the organ, and then having the full cost of the transplant on top of it, that's just a tough commercial business, right? That's just, you don't have a lot of opportunity to make money on top of the cost of goods, the COGS associated with that therapy. And for us, because we're using these discarded organs, we're isolating them in a relatively straightforward way, at least relative to some of these other technologies. And then we can divide those, even those relatively minor costs over so many different patients, right? Just the raw mechanical business of this is, I think, extraordinarily compelling, and really at a time when cell therapies are struggling that way, right? A lot of cell therapies are struggling precisely because when they're tailor-made for each individual patient, there's just no way to avoid a lot of those costs, right?
Yep.
And so they end up being both very expensive, because even then, just the base COGS are so great, to make any money over and above that, by definition, you have to charge more, right? So I think when you look at the average liver transplant costs right now are about $1 million for the acute in-hospital charges, right?
Yep.
So, that's a very high bar. Now we're talking about, we're going to be able to develop and deploy, and the endoscopic ultrasound. Sorry, that was the other thing, right? Endoscopic ultrasound, just as a procedure, it's billed, the CMS code is about $2,500. It takes about 10 minutes for an endoscopist, and we use standard, Olympus equipment. We use kind of the standard EUS equipment, because we didn't want to introduce a novel medical device, because again, that's just nothing but complexity. So we're using a lot of standard endoscopic equipment. And because the endoscopists are familiar with the surgical engraftment procedure, they don't do this procedure today, but it's new in terms of engrafting the lymph nodes. Well, what they do is a similar surgical procedure to look at these same lymph nodes and the spread of pancreatic cancer. So they're used to using the endoscope to going transluminally through the gut wall into these lymph nodes. Usually they retract some tissue to look for metastatic spread. In our case, they're using the same procedure, the same tools they use, and they're engrafting about 1 ml of our cell therapy into those same lymph nodes. So again, we've just decreased the friction to deploy this clinically. And I guess the last thing I'll say is, having gone through FDA approvals and commercialization of therapies before, when you have to field a large sales force, right? It's an enormous capital drain. I mean, and how many companies have made the mistake of either trying to do it themselves, and that's why you end up so often partnering with large pharma, right? They have the existing sales force, and from a capital resource drain, I mean, talk about a giant sucking sound. I mean, it just, when you try to do that yourself and you're detailing a few tens of thousands of docs, you need this massive sales force. Doing what we're doing with patients with no other options, who are desperate for novel interventions. You can have a handful of academic centers processing these cells, will service the entire continental US. And so you don't need to field a sales force of thousands to spread the word about this therapy, right? And endoscopists and endoscopy suites that hospitals have already invested in, this is another source of revenue generation for them. Right. So you're always trying to look, again, in this very unsexy way of just what are the business mechanics of making this a compelling business, because ultimately, right, that's how capital flows into the area.
Yeah, I mean, it's so elegant that, you've from the early stages thought through all these issues, because most people don't. Right. I mean, most people don't think about this stuff that early on. And, right. I mean, thinking of the COGS and so forth at this stage might not be sexy, but hey, you're either going to have a product or not. Right. So, I mean, you have to. And so I once again, I think it's extremely elegant that you've, and that's why I was, I enjoyed watching those other presentations as you've thought through this entire process. So no, it's spot on. So moving from, Ligenesis, which is an extremely sexy story, to another equally sexy one. And I'd say, I've had Mike Levin, well, actually on three episodes too on this platform and a previous one. So I know Mike pretty well. And, saw Morphoceuticals coming years beforehand, but nonetheless, I'm glad to see it here. And here we enter the amazing world, of sort of all the cool things that happen in the biophysics domain, so the membrane potentials, the forces of mechanotransduction, all the stuff that Mike has always referred to as sort of the information that sits above the genome. He looks at the genome as really hardware, and the hardware is played by all these other cool forces that we don't really think as much about as we focus a lot on the cell and the DNA nowadays. This is not a typical sort of electroceuticals approach. You're focusing more on the ion channels with traditional drugs as opposed to some other entities. Walk us through, I know because they're early stages here, but nonetheless, some of the coolest biology I think of. Take us through sort of a little bit where you're going with Morphoceuticals and what's happening.
Yeah. So, like you, I share an incredible enthusiasm for the science that that Mike Levin, his colleague David Kaplan from Tufts have advanced into Morphoceuticals. And really hats off, by the way, to Alex Pickett, who's a managing partner at Juvenescence. It was Alex, I think, who originally saw the technology and kind of realized its commercial potential. So, I mean, Juvenescence is doing exactly what they do well, right? Is look at those early stage technologies for their commercial potential. So kind of hats off to them really for recognizing it. But yeah, Mike's approach to really understanding this kind of software of life that he talks about, right? This higher order, these connections among cells that are fundamentally ion channel and gap junction modulators, ways to exert changes to that programming to really produce remarkable therapeutic effects. So you've had Mike on, folks that are familiar with his work. Certainly the recent publication on in regenerating the fully functioning innervated frog leg in an animal that doesn't naturally regenerate. We're now extending that work to mice. Into mammals, right? Because you want to move that forward again from an FDA perspective. That is incredibly exciting biology. It really is an honor to be associated and partnered with it. And again, really what I'm doing there is like, you know, I read recently, I forget where, I'm sure you've heard it before, this distinction in early stage startup between artists and soldiers. And you know, Mike is truly an artist. I mean, he has a comment about his Picasso frogs, right? Because he can get frogs to grow, you know, arms out of heads. And, but really, Mike and David really are artists. That is, you know, they are incredibly, staggeringly creative. And working at this, as so often happens, right? I'm sure you've seen this, at the intersection of fields. They're at the intersection of, you know, Mike has this kind of computer scientist view of biology combined with this electroceutical ion channel. And it's at this fascinating intersection of fields where his work resides. And ultimately, though, to kind of turn those things into commercial products, which also means bringing capital in to help fund the research to advance the commercial opportunities, you really need soldiers. And so I'm really that soldier that way, insofar as like not in a macho sort of way, in a sort of like you understand an operational objective, what it takes to go from where you are today to achieve that operational objective. And then it's kind of working with the artists on that early stage technology to start, you know, guiding some of the scientific studies so that instead of just being exploratory, they start to check some of the boxes that the FDA wants to see, again, in terms of safety and efficacy and dose ranging, and things that aren't necessarily the things that get you into nature, right? And that's often the tension in academia is the things that get you advancement and professional acknowledgement in terms of publications in Cell Science and Nature. That's the pinnacle of academic achievement. From a regulatory perspective, it's just the start. All you've done is really discover, which is obviously incredibly important, right? It's like the genesis of all of this. But ultimately, to advance it to commercial therapy, you have to do a lot of kind of, again, unsexy work, a lot of dose ranging work, a lot of genotox work, carcinogenicity. Like you need to just understand what you're doing. Now, Mike and David's work has an additional very valuable component, which is, you know, he is also able to leverage a lot of existing drugs. So there's both a novel drug discovery opportunity here that's really quite profound, and he's already demonstrated that. But it's also the case that for some of these cocktails, you can use existing FDA-approved drugs that have other indications because, you know, they affect ion channels. So obviously, a lot of neurology, psychiatry has been you know, playing around with ion channels for a very long time. But the realization that that the modifications to those ion channels in a way that we're used to thinking in a CNS way can also be used to exert really profound therapeutic effects by altering the software of life in the morphogenic space of altering anatomy. That, again, is this incredible creative artistic achievement. And now what we're doing at Morphoceuticals, right, is now trying to think about, okay, having established that we can do this, what patients can we treat? Where can we engage regulators in that discussion, whether it's amputation, stump health, right, as low-hanging fruit, where like a lot of orthopedic surgeons will tell you that when someone has an amputation, a huge predictor of quality of life, of whether they're going to be able to eventually use a prosthetic, is how well the stump heals. And again, it's very sexy and cool to think about limb regeneration, and that's absolutely where we're going at Morphoceuticals. the low-hanging regulatory fruit may not be full-on limb regeneration. It may be something as straightforward as amputation stump health, where when people have an amputation, you help make sure that that stump is well vascularized, that you don't have a lot of neuroma formation. That's a way in which you can affect thousands of lives in a pretty straightforward way from a regulatory perspective, and then build on that for some of the other remarkable, whether it's fingertip regeneration, whole limb regeneration, that is absolutely now a tractable problem thanks to the work of Mike and David. But turning that into an FDA reality, freeing the capital to come into it so that folks see it as a viable commercial opportunity, that's sort of the unsexy business of soldiering it from the creative genesis in an academic lab, out into its early startup phase.
Excellent, really excellent. Mike, in the bio, I mentioned, you've, aside from being involved in all these areas, you've had experience at the agency, small molecules, biologics, drug delivery systems, and there's now here we are in 2022. Obviously, we talk about regeneration and repair, and they're also under this umbrella. Juvenescence has sort of this core aging focus, and anything with regeneration and repair ultimately feeds into that. I'd like you just to say a couple words about two things, just to get your views on this. Number one, as we talk about some of these novel interventions, like say Morphoceuticals, where you may, as you were saying, even if we're dealing with repurposed drugs, you may require a little cocktail of stuff to ultimately regenerate that leg. And then obviously Ligenesis has its own unique, we haven't seen things like this before. Talk a little bit about sort of, how that sort of impacts the way you think and you feel about going into the agency with some of these complex, novel interventions. And then if you could also say a few words, because I know it's a debated topic, it's sort of in the social sphere, but this whole theme of aging as a disease and ever getting FDA to say, yes, you can develop a drug for aging versus, these, liver regeneration or whatever it may be. Just say a few words about that and sort of any involvement you've had to date with sort of that whole discussion debate.
Yeah, sure. So to take the first part of the question in terms of engaging regulators on highly innovative therapies, it's very easy to bash the FDA, and it's not that sometimes they don't deserve some criticism and some kind of corrective input on some of the way that they approach drug regulation. I think that's more than fair. That being said, like we've been working with CBER and the Office of Tissue and Advanced Therapies at Ligenesis. They're used to seeing incredibly innovative stuff, right? I mean, these are folks that see a lot of cutting edge innovation coming out of academic labs. And I think one of the things that has benefited, I think, Ligenesis, and we have an excellent regulatory consultant group that helps with this as well, is to appreciate that they see a lot of things coming out of academic labs that maybe really aren't ready to have an IND and to go into people because folks haven't done the kind of foundational work to establish the safety and efficacy of that. So I think when you engage the regulator with a history of drug development and understanding what the whole process looks like all the way through to approval, and when you're trying to get an IND on some of these innovative technologies, appreciating that they're always looking out for how are you going to hurt someone, right? It's as a company, like you're always thinking of unmet need and how am I going to help these patients? Regulators are concerned about, are you going to inadvertently hurt someone? And so if in your regulatory package, you can tell a story that makes clear you are thinking of safety first, and you're being very cautious and programmatic and pragmatic when it comes to safety concerns. And yes, the sexiness you kind of sometimes want to lead with, and especially on an investor perspective, that is what you lead with. That's not how you lead with regulators, right? You lead with regulators in a very cautious, patient safety first kind of perspective. And I think that buys you a lot of credibility because it provides reassurance that you're putting patient safety first, not just the fact that you have this amazing discovery that could one day do X, Y, or Z. So that's one. I guess on the second comment, I should preface these opinions are my own on the aging space. I guess I have maybe somewhat of a contrarian opinion on some of this, maybe. look, there's been a huge renewed interest in anti-aging and framing aging as a disease. And in many ways, obviously, part of me hopes folks are right that way, and that it ends up eventually being tractable, that is aging itself. I personally am skeptical that today is the day for that. I think there's a long way to go. Mm-hmm. That being said, investments in this space, looking at things like regeneration, or you just look at the other tractable indications between here and aging. I guess part of my thinking is that just looking at selfishly as an investor and return on investment. Now, what kind of multiple on your investment can you realize by investing in this space? Yeah. I would argue that a better investment is in technologies that have existing FDA indications with known patient groups that are treating those patients, that there's a lot of fruit to be harvested here, both in terms of medical benefit, but also return on investment, mm-hmm. before you ever crack the nut of aging and getting the FDA to acknowledge that aging is a disease.
Gotcha.
I think that is going to be a years or decades long push. We might get there, but I think in terms of de-risking an investment, man, whether it's autoimmune diseases, all the inflammatory diseases, the entire field of oncology, right? All the regenerative medicine possibilities that you see, there's just so much wealth of fruit to be harvested on more tractable indications without the kind of esoteric debate of aging as a disease, and there's going to be this single mechanism to treat that. Time will tell whether or not this is kind of a fad that will pass. But I think in terms of an investment that you can realize a huge return on is in technologies that might one day address aging. In fact, I mentioned the thymus work we're doing at Ligenesis, right? Mm-hmm. But way before I personally would engage the FDA in an aging conversation, I would engage them in an autoimmune disease conversation, or mm-hmm. I would engage them in a disease around cancer prevention, as hard as that is, yeah. before I would engage them in an aging. And if you're right, you're going to have a huge return on that investment without having to kind of chase the golden chalice of aging itself.
Gotcha. I appreciate your insight on that as well. Yeah. That's awesome. Mike, I would like to just for a couple of minutes, if we could, transition to your historic work at harm reduction therapeutics, because two reasons. One, so I'm sitting here in the downtown city of brotherly love, Philadelphia, Pennsylvania. Yeah, we are known for the cheesesteak, the soft pretzel, the Liberty Bell, and the largest open air drug market in the United States nowadays. So we got a major opioid problem up here. Yeah. I'd like if you could just say a few words about your historic, because this is a nonprofit drug company that's involved with an OTC switch with naloxone. If you could just say a few words about that, but then also, the RX to OTC path has been, we don't hear as much about it as I think I used to hear much more about it back in the 90s. Any interesting things happening there in general? Because there are, there's this always sort of a debate to where certain moieties may drop, whether they're technically drugs nowadays or whether they're OTC products. Some of these unique interventions that are out there. Absolutely. I'd love to just get your feel for some of that, the RX OTC space, and maybe why we might not see more happening there. Maybe we should, in terms of some of this repurposing. Love to get your thoughts on that.
Yeah, space. No. Well, one, thanks for your interest in asking about it. So Harm Reduction Therapeutics is a nonprofit 501c3 pharmaceutical company that I co-founded with a colleague of mine, John Penny, about 5 years ago. Our sole purpose is to take a low-cost or free over-the-counter naloxone intranasal product and make it available over-the-counter. To your point about the problem there in Philadelphia, right? The opioid epidemic continues to kind of wash across America. 188 people die every single day of opioid overdose. I was actually involved, my family and I, in an overdose reversal in New York City on vacation just this past summer. So it is absolutely something that can affect anyone at any time. And naloxone is a tragic case study, right? Here's a drug that is incredibly safe, highly effective in reversing opioid overdose, approved by the FDA in 1971, off patent since 1985.
Yep.
And as the opioid epidemic unfolded, the price of naloxone skyrocketed, right? And so markets do what markets do. As demand rose, so did the price. But it really was a failure of, I think, our current patent laws, where folks would run to space, right? They would create intranasal formulations, they tweak this or that, get a patent on it, and then use that to increase the price, right? So that 4 cents of a life-saving dose of naloxone would sell for $140 in a case of intranasal sprayers, or an auto injector that will remain unnamed was selling for $4,500, right? And so I really saw that as really kind of a crisis of conscience for us as a drug development industry. Yeah. And the FDA, the CDC, the American Medical Association had urged pharmaceutical sponsors to take their products over the counter. 5 years ago, we approached a number of them. None of them said that they were intended to do so. And so, frustrated with that, we licensed our own intranasal formulation. We reformulated it as a slightly higher strength than it was existing, and actually just submitted the NDA for the product. It's now called Revive. Nice. It's a 3 milligram intranasal naloxone spray. The new drug application is now under review. We submitted the full NDA just a couple of weeks ago, so your question's very timely. So we asked the agency for priority review. Hopefully we'll get a 6-month review rather than the standard 10-month PDUFA review. If approved, we would commercially launch in early 2024. And the goal will be to make free, low-cost naloxone really available to everyone. So our plan is to manufacture millions of doses per year to try to make a difference in the ongoing opioid epidemic. The dual tragedy, of course, right, is when you look at it from a macroscopic perspective, what's happened? Heroin potency has increased. Yep. Its price has decreased. And the retail distribution is as robust as it's ever been. You can find heroin in this country as fast as you can find coffee if you're looking for it, right? I mean, the retail distribution of heroin is staggering. Now it's contaminated with fentanyl. It's almost impossible to find pure heroin now. The heroin supply chain is largely contaminated with fentanyl. You see fentanyl pressed pills, right, that appear as prescription drugs, and especially teens who access a lot of those pills through Snapchat and what have you, having fatal overdoses. In fact, a recent survey found 40% of those drugs contained a lethal dose of fentanyl, right? And so in the context of all that, what's happened with naloxone? Naloxone, the price has increased. It stayed as a prescription drug, right? And so what the purpose of harm reduction therapeutics was to decrease the price and make it more widely available as an OTC. Hopefully that will happen. The FDA has certainly called for it to be OTC. They even developed the draft label for it, which the FDA has never done. So like, credit where credit's due. They published in the New England Journal of Medicine a couple of years ago. They did all the work that usually is on the pharmaceutical sponsor to do the drug facts label, to look at label comprehension, to make sure that people, because that's the key regulatory threshold of RX to OTC, right? You have to make sure that, of course, the drug has to have a very pristine safety profile, but you have to be able to show that the average layperson understands when to use the product and can use it correctly. And that can be a very high bar. When you do those, what are called actual use studies, you typically recruit about 30% of what's called low medical literacy lay people to see if they can understand and use the product. And that's not easy, right? When you look at the US population as a whole, making sure that something is easy enough for literally anyone to use is actually a high behavioral hurdle. And so you're absolutely right. There's this field of regulatory science in RX to OTC switch. And I also think you're right that field, I think, was much more active in the 90s and early 2000s. You saw, all the nicotine replacement therapy products. You saw allergy meds, asthma. You saw all these things switching. There are still folks that specialize in that area. Penny & Associates, that's the pharmaceutical consulting firm that John Penny co-founded, is still kind of state of the art in that field. I think the agency has recently released new guidance documents trying to encourage that maybe there's a way to get more consumer lay access to drugs. Maybe they aren't fully OTC, maybe they're RX, but if you can complete like an online tool, or if there's some way to document that you in particular can use the product appropriately, maybe there is a way to access drugs. So there's been some movement trying to find novel ways to allow lay people to, consumer access to some RX products. But the regulatory hurdle is one that a lot of people don't appreciate how challenging it is. That you need this combination of a pristine safety profile combined with being able to have folks that are not medical experts recognize they have a condition, understand how to use a product, when to stop using the product, right? There's a lot of onus on the lay person when it comes to consumer healthcare products. Circling back to naloxone, it is a very unusual consumer healthcare product insofar as there are very few that are acutely life-saving, right? So that's one where you really don't want to get it wrong. And in fact, back to your about product placement, this is actually what the Revive will look like. Which for your folks on the podcast is not going to be helpful at all, but it's very simple intranasal sprayer, single-use device. But it needs to be that, right? Otherwise, the average person won't be able to save a life when it comes to it.
Really awesome. No, that's great, Mike. Very impressive. What while I have you, anything else hot coming up for the remainder of 2022 as we go into 2023? Obviously, you have the NDA coming up for harm reduction. Some fundraising rounds with the other companies. Anything else you want to mention while I have you? Or 10 new companies you're starting. Whatever. Just please take the floor way out.
Well, look, we are starting additional clinical sites for our phase 2A trial at Ligenesis for end-stage liver disease. So we hope in the coming weeks to have some announcements about patients being engrafted in that trial. So obviously very excited about seeing the first patient engraftment in the phase 2A trial at Ligenesis. So that's going to be very exciting. We will have a round of capital that we'll be raising probably in the middle part of next year for Ligenesis that will see that phase 2A trial through to completion, as well as advance some of our other opportunities in pancreatic islets and diabetes in particular, but some other technologies as well. So yeah, we will be raising a round of capital at Ligenesis. For Morphoceuticals, we're actually just weeks away from announcing that we have raised a seed round of capital at Morphoceuticals. Very excited about the well-known VC firm that's leading that round, with Juvenescence continuing to support in a very significant way that round as well. So as soon as that round closes, which hopefully will be just a few weeks from now, hopefully less than that, we'll have a press release around the seed round at Morphoceuticals and really be off to the races there. And then, yeah, fingers crossed on the FDA acceptance for review and hopefully priority review on the Revive, the intranasal naloxone opportunity.
Awesome. And I'm rooting you on for all of this because
thanks so much, Ira.
It's so impressive, and the integrated thinking that you bring to this, Mike, is just as I said, it's very refreshing and elegant to see. You talk through these studies and these situations in this detail. So really very impressive. For everybody that's going to be listening across the various podcast networks to this episode, or of course watching on our YouTube channel and blog, again, you've been listening to Dr. Michael Hufford, co-founder and chief executive officer of Ligenesis, interim chief executive officer of Morphoceuticals, scientific advisor, Juvenescence, co-founder and CEO of Harm Reduction Therapeutics. We'll put links to all of this in the bio of the show. But Mike, I want to again thank you for taking the time out of your schedule to come talk to us about all these topics. Obviously, thank you for doing this. And as we say on our show, thanks for helping to create a better tomorrow through what you're doing to so many people. Really, really great stories, Mike.
Oh, thanks, Ira. I appreciate it.